As the thick and thin filaments in the sarcomere slide past one another during contraction, force gets generated. In other words, muscle contraction generates mechanical force. And that's important because it's that force that we can use to do work with our muscles. It turns out that the mechanical force generated is relative to the number of myosin heads that are coming in contact with the thin filaments. The more myosin heads are in contact with the thin filaments, the greater the force that's generated. It's important to note that even at rest, even when a sarcomere is completely relaxed, there are still some myosin heads overlapped with the thin filaments. Not many, but some. In other words, at any moment, some of those myosin heads could reach up and contact the thin filament. When contraction begins and the thin filaments start to slide, more and more of those heads will be overlapped with the thin filament. And when they contact the thin filament, more and more force can be generated. Something else to note is this. Once contraction begins, once a sarcomere begins to shorten, it will shorten maximally. The difference that's generated during muscle contraction in terms of force is not within the sarcomere. In other words, the sarcomere is going to contract maximally. What differs between a smaller amount of force and a greater amount of force is how many sarcomeres contract maximally. The more sarcomeres contract, the more force is generated. You can also see variations in how many myofibrils are involved in a contraction. So, more myofibrils contracting will also increase the force of the contraction. You can also see variations in how many muscle fibers are involved in the contraction. So the more fibers are involved, the greater the force generated. Once a sarcomere starts to contract, it will contract maximally. But force generation can vary because of this phenomenon that we call recruitment. The more sarcomeres are recruited, the greater the force. Once all of the sarcomeres in a myofibril are involved in the contraction, well, you can involve other myofibrils. And once all of the myofibrils are involved in contraction inside one muscle fiber, you can involve other muscle fibers and increase the force that way. So there is a large range of how much force can be generated during the contraction of a single muscle. Let's take a look at a sarcomere both in its relaxed state and in its maximally contracted state, and think about force generation. Up at the top on the left, we're looking at a drawing of two sarcomeres side by side. These sarcomeres are in a relaxed position. The Z lines are separated from each other. These sarcomeres are wide open. They are in a relaxed state. Notice that even in this wide open, relaxed state, there are still a couple of globular heads overlapped by thin filaments. They're not contacting the thin filaments right now, but they are positioned so that they could contact those thin filaments. There are lots of other heads that are not overlapped yet by the thin filaments. So even at full relaxation, there will be some overlap between globular heads and thin filaments. Now let's start contracting. Let's start these globular heads binding to these thin filaments and sliding them towards the center of the sarcomere. Same thing's happening over here. These heads are binding to the thin filaments and sliding them this way towards the center of the sarcomere. Once that begins, this is going to happen. These sarcomeres are going to shorten maximally. So at this point, all of the globular heads are lying underneath a thin filament. Only some of them are overlapped in relaxed state. All of them are overlapped in contracted state. Now, take a look at this graph over here on the right. This is a graph that we call a length tension diagram. On the x-axis, we're looking at the length of the sarcomere. Now, sarcomeres are microscopic. This is in micrometers. On the x-axis, we're looking at the amount of force generated as a percentage of maximum. So it runs from 0% of maximum all the way to 100% of maximum. Now this graph is a little strange in that if you want to read the line on the graph, you have to begin all the way over here because this is at 0 tension, fully relaxed. So the sarcomere is at its longest, its most wide open at this point, something close to 3.5 micrometers. Now, follow the red line. As the sarcomere gets shorter and shorter and shorter, tension is building, force is building. As the sarcomere shortens, more and more and more force is generated until we hit a peak. There's a little bit here of a plateau. It's not very long, but we have hit maximum force generation. And that maximum point comes when that sarcomere or those sarcomeres are maximally contracted, maximally shortened. They're showing us that same thing up here. Here's the sarcomere completely relaxed. That's happening right here. As that sarcomere gets shorter and shorter and shorter, as it contracts, force goes up, 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 till it plateaus. These sarcomeres, once they're completely contracted, they're generating as much force as they can. Now, if you tried to shorten the sarcomere past this point, if you tried to shorten these sarcomeres beyond this point, you could do it, but you would damage the sarcomere. Because remember what's happening at this point. These thick filaments are bumping into the Z line. Now remember, there is a protein in between here that we can't see, that titin protein, along with other proteins, accessory proteins. But essentially, the thick filament has encountered the Z line. If you pushed past this point, if you forced this sarcomere to get even shorter, you would damage all of this accessory protein and you would damage the sarcomere. At that point where you're forcing the sarcomere to get even shorter, you're losing force. You're damaging the sarcomere, you're losing tension or force. So there is a peak of force generation, and that's when the sarcomere is maximally contracted, but not to the point of damaging the structure. That's called a length tension diagram. On this slide, we're looking at several people who are flexing their arm, flexing their biceps muscle and other muscles in their arm. They are contracting that muscle and shortening the angle between their forearm and their upper arm. I put these pictures in because I wanted us to think about the fact that in human beings, even though we all have the same muscle groups and the same individual muscles, the size of those muscles really varies. It varies not only between the sexes, between males and females, but it varies within the sexes, right? We think about males as having larger individual muscles in general. That's related to sex hormones and secondary sex characteristics. But we also know that within a sex, like within the male, there's a great deal of variation in the size of individual muscles from person to person. It's important for us to think about why this is. Now, obviously, there are ways to make your muscle mass increase that involve illicit drug use and things like that. I'm not talking about that. I'm talking about how ordinary exercise and working out can increase the size of muscle mass. It's important for us to know, as students of cell biology, that a person who has a larger muscle has not increased the number of muscle cells inside that muscle. This man does not have more muscle fibers than this man does. What this man has done is increase the size of each individual muscle fiber. And he has achieved that by increasing the number of myofibrils inside each individual muscle fiber. Muscle cells, muscle fibers, like other types of cells in the human body, do not increase in number once we reach our fully grown adult body. Neurons are like this too. Muscle cells are placed in the body, a certain number of them, and then that number is set. Now, it doesn't mean that those cells won't be replaced over time. They will be, but those cells are not going to start dividing again, increasing cell number. We are not genetically programmed for that to happen. It's not that there aren't stem cells in muscle, there are, but they are not transcriptionally active. Once all of your muscle fibers are in place, they no longer divide in your adult body. Now that seems counterintuitive because we know we can get our muscles to be larger with exercise. It's just important to know that you're not increasing the number of cells in an enlarging muscle. You are simply increasing the size of each muscle fiber when you exercise. And the size is getting larger because the number of myofibrils is getting larger. And the number of myofibrils means that there are more sarcomeres inside of each muscle fiber. And that means more force can be generated inside of that muscle. Remember, the more sarcomeres that are involved in a contraction, the more force you can generate. So having more sarcomeres available means that that muscle can generate more force. So we've said that once contraction begins, the thick filaments begin to bind to and slide the thin filaments towards the center of the sarcomere during this sliding filament contraction. We've also said that when a thick filament contacts a thin filament, when the head of a thick filament binds to a thin filament, we call that a cross bridge. We said that cross bridges form and then they dissociate. And then they form and then they dissociate. because each of the sliding motions that occur during muscle contraction are relatively short-lived. The distance that the thin filament slides each time the head moves, that distance is relatively short. So cross bridges are transient. The head will bind to the thin filament, slide it a little bit, then the head will let go. It will bind again, slide it a little bit more, and then let go. This binding and then releasing has to happen really fast, and again in a coordinated manner. And as you already know, this motion, this motor protein activity requires energy, and that energy comes from ATP. Myosin heads can cycle on and off of thin filaments by changing shape. And that shape change is brought about through the energy of ATP. We talked about how ATP is required for the movement of kinesins. And remember, we said that just the act of ATP binding to the globular heads in a kinesin causes those kinesin heads to change shape in that neck linker region. We also said that the globular heads themselves caused the ATP hydrolysis. The same is true in myosin. The myosin globular heads have intrinsic ATPase enzyme activity. When the ATP binds to the globular heads, there is an enzymatic area in that head that can hydrolyze the ATP, converting it to ADP and phosphate and a little bundle of energy, creating the shape change that allows that globular head to slide the thin filament a short distance. On this slide, I've outlined the step-by-step process that's occurring as a sarcomere shortens, as a sarcomere contracts. You could jump in at any moment in this cycle of contraction, but we're going to jump in at the moment that the myosin ATPase enzyme, in other words, the enzymatic region of the myosin head, is going to split the ATP into ADP plus phosphate plus energy. So ATP had already bound to the globular head, and the globular head just split the ATP and released a little bundle of energy. What's going to happen in that moment is that that globular head is going to cock backwards. Think about how you cock a gun, how you reach up with your thumb and you cock back a gun. The same thing is going to happen with the myosin molecule in that the head is going to get into what we call a high energy conformation because it's cocked backwards. Now the next thing that happens is the phosphate group will come off of the globular head. That's going to cause a shape change, and that will allow that head to reach up and form a cross bridge with the thin filament. Then the ADP will be released from the globular head. That's going to cause another shape change, and that's going to allow that important power stroke to occur. Just like the power stroke we talked about with cilia, the globular head and the heavy chains of the myosin, they're going to force that thin filament to slide towards the center of the sarcomere. And I'll show you a picture of this in just a minute. Now we're in what we think of as the low energy conformation. The energy that myosin molecule got from that ATP is gone now. But that myosin head is still bound to the thin filament. It just doesn't have any more energy for motion. we said that cross bridges have to form and then break and then form and then break in order for this to work. So something has to happen to allow the myosin head to let go of the thin filament, and that something is more ATP has to come in and bind. That's going to cause a shape change, and the globular head is going to release the thin filament. Now, the enzymatic activity in the globular head is going to split the ATP, and the head's going to cock backward. And we're right back to the beginning of this process. The phosphate gets released, the cross bridge forms. The ADP gets released, the power stroke occurs. Now we're in that low energy conformation, so more ATP has to come in and bind. The head's going to let go. The ATP gets hydrolyzed, the head cocks back. And it goes on and on and on until the sarcomere is maximally contracted. The next several slides show this process step by step in pictures. So we're at the level of a single sarcomere. We're looking at a thick filament in brown and a thin filament in blue. Most of the thick filament has been removed. We can only see one globular head here at the end of one helical heavy chain. This globular head is in its high energy conformation. It's cocked backwards. It's bound to ADP and inorganic phosphate because this globular head has just hydrolyzed a molecule of ATP, and the energy was released and it cocked back. Now, the phosphate group is going to get released. The cross bridge is going to form. That cocked head is going to go up until it contacts and binds to the thin filament. Next, the ADP is going to be released. Start up here. We've just formed a cross bridge. Now the ADP is coming off. And what happens? We get that power stroke. We used to be sitting right here, bound to the filament here. Now we're going to slide forward and push that filament forward towards the center of the sarcomere. Next, we're stuck. We've moved that thin filament a short distance. We're still bound to that thin filament. We're still in a cross bridge, but we're out of energy. What do? Well, another ATP comes in and binds. And that's going to allow the cross bridge to break. And now we're in a very low energy conformation. Here we are down at the bottom. The new ATP has bound, the head has let go, the cross bridge is broken, but we're in a low energy conformation. We can't move. So, hydrolyze the ATP, head cocks backwards. Now we're in a high energy conformation. We've got potential energy, we're ready to move. So let's put it all together. The ATP has been hydrolyzed. The head has cocked back into a high energy conformation, ready to move. The phosphate group is released. The cross bridge forms. The ADP gets released. The power stroke occurs. More ATP binds. The cross bridge breaks. ATP gets hydrolyzed. Head cocks back. Phosphate gets released. Cross bridge forms. ADP gets released. Power stroke occurs. More ATP comes in. Cross bridge breaks. Whole thing just keeps going and going and going until the thin filaments have been slid towards the center of the sarcomere as far as they can go.